Thank you. And thank you for asking me to do this, which is a great pleasure. Let's quickly go over Giulio's CV. Giulio did his PhD with Meinrad Buslinger at the University of Zurich in 1991, and then came here to EMBL as a postdoc with, where he worked with Giulio Draeta and Sarah Cordnish, who I believe also won this award. He then, after his postdoc, stayed on as a staff scientist until 2004, with a brief stint in between as guest professor at Bologna. And during this time, he also founded CellZone. In 2005, he became guest professor at the Medical University in Vienna and founded the CeMM, of which he's been the director since. Not having founded, being founding director and established enough institutes and institutions, he last year agreed to become the founding director of yet another institute in Palermo, where I'm sure we'll all be very happy to visit him. He's also been member of the ERC Council for a few years, and he's been on loads of committees and boards, including, as we've heard, the Alumni Association, EU Life, and many others. And of course, he's a very successful scientist with tons of papers, patents to his name, 2 ERC grants, etc., etc. So far, so good. That's the bare bones. So he's just a normal scientist, like the rest of us. Well, actually, normal is the last thing that you would call Giulio. And in fact, if you ask people, they say things like, Giulio, oh yes, what's special about him? What's not special about him? Maria, how long do you have to listen to me? Or he never settles for the ordinary. He raises his groups to new levels. So I think all these things are absolutely true. And in fact, what I thought of when I thought of having to talk about Giulio is a quote by Andrew Geim, the Nobel Prize winner for graphene, who said, "It's better to be wrong than to be boring." Giulio, that's you. But that of course implies he's sometimes wrong. So, and I'm sure he is, and I'm sure many of you know examples, and I won't dwell too much on this, but again, if you ask people, you get quotes like, good things he wants to do sometimes just break the furniture. Or, some things come out of his mouth really wrong. So he's politically incorrect, he's not deferential, he's quick and fast. But the nice thing about Giulio is that he's also the first one to admit that he's wrong. And he's the first one to go and eat humble pie, apologize, and talk to people and correct it if he feels he really was wrong. And I think this is something to do with something he said in an interview, and that is that he loves people. And I think that enables him to do this, to just go up to people and interact with them in the way he does and in the way that we all love. Politically incorrect, let's return to that too. I mean, you know, it's really amazing because he is also the politician's poster child for what he's done in terms of innovation. I now have to endure Brussels, and I have to hear this call for innovation all the time while poo-pooing fundamental science. You've just been fantastic in doing both without compromising the value of fundamental science. Really admirable, and just another facet. So Giulio is scintillating, all over the place, loves everyone, is loved by everyone, but he's also demanding of others in his request that everybody think big. And I had to actually experience that myself when he invited me to a conference. It was the 10-year anniversary of EU Life, where he asked me to be on a panel, and he wanted everyone to think big, dream big about the future of science. So we had a little Zoom call beforehand with the panel members, and some of them did think big. And I sort of said, well, Giulio, I could say this, or I could say that. He said, no, I mean, come on, think bigger. I said, well, how about this or that? Maria, come on, think of something. I got really scared of him because he started putting me down. And so then I said, yeah, but Giulio, I actually think the conditions for doing research that we have here right now aren't so bad. He said, Maria, you can't say that on this panel. You are so conventional. This won't do. Well, I tried hard. I don't know if I live up to your standards, Giulio, but one thing is clear. We all love you, and we all think you really deserve this prize. So, over to you. Oh, wait a second. I actually forgot to, one of the things he does at conferences and meetings, he makes people wear these big signs. And here's one that he wore around his own neck, where he describes what he wants, chair of the board of this or that, what he likes, protein complexes, and describes himself as pompous and self-righteous director of CEMM. So he has some humility too. Anyway.
Thank you so much.
Come on.
Thank you.
Very nice.
Yeah.
What do you want to give him? Medal? Give him medal. I'll give him the.
So we need to be in front of
Yeah,
the.
we will
How does
pass
this
it to you, and then we take a picture.
Els Nush
I
will-
will
Oh my
just
God.
mention that the 2024 Lennart Philipson Award goes to Giulio Superti-Furga, because none of us has said.
None of us has
Yeah,
said
this is very professionally organized award ceremony.
Thank you so much.
I think
Everybody will do you. Go left and right. I need to submit. Okay, thank you so much. This is lovely. Wow. Just as I was happy that I got Maria, not Pavel, to give a talk, it turned out Maria is far worse. Better. Okay. Okay, I'm between you and the celebration, and I'm between you and the soccer game, so I'll try to be concise. It's true that I love people, and I wouldn't say I learned it here, but pretty much I did. And it's true that I love EMBL. So what I'm going to tell you tonight is just it's a sentimental, romantic love affair with EMBL. Maybe this is a little bit banal. Some among you will wish I would be a little bit more formal. But let me start by saying it's an incredible honor to receive this. Imagining being hanged on that wall along with some of the heroes is really humbling. I am absolutely also flabbergasted by the fact that this award coincides with this incredible anniversary. So I'm really part of this celebration. And I am just here as one example, like Thibaut, who I would like to congratulate. But other people, we are on the shoulder of giants, and more people will come. This is my community. I've never been feeling more home scientifically than when in this square kilometer or so. It's almost inexplicable. And certainly we all have seen in these days the amazing science that keeps being generated in this place and its different outstations. And I would like to thank Edith and thank all the members of this community for keeping us together as a community, and then also the Alumni Association, and inspiring us. In fact, it's really Edith that has a role to say about this, and I will see it in a minute. It's a long love affair. So 40 years ago, I came as a 22-year-old student to this conference. Maybe Thomas was there. I think nobody else probably in the audience was there. This was an incredible conference on transcription. Still, we didn't have an Operon, so it was still in the Max Planck next door. And I remember absorbing each and every word that was said, being incredibly impressed by the jeans jackets and the long hair of Erwin Wagner and Uli Ruther, who climbed on stage. I know, I must have shown this before. These are these hippies coming on stage like this. Should I really come to this panel? And then they sit down. And Erwin sat down like this. And I said, wow, this is the guy I want to work with. And I did apply to his lab later, but then other things happened. So it's a love affair that is long-lasting. Then when I did get the chance is because Giulio Superti-Furga, who was an incredibly inspirational person, and to this day one of the best translational oncologists we have at MD Anderson, and juvenile in his criminal energy, I don't know whether you met him recently, had an incredible lab, assembled a lot of very, very good people. We were in a special lab, and I really enjoyed every minute. The only problem is that after a year, Giulio decided to move on and start a company in Boston. So we had to think about something else. Here I was briefly thinking that I would, I came as a first molecular biology expert in the Drezza lab. So I started doing human cDNA libraries to put into Pombe. And every week I was trying to complement a nice Pombe mutant, cell cycle mutant, CDC2. And every night I was thinking, my God, I have all these clones growing, and each one of them will be an incredible success. And I did find a lot of interesting things, but what really did happen is that I started expressing oncogenes in yeast, human oncogenes or chicken oncogenes in yeast, in fish and yeast. And that turned out to be quite interesting because it allowed me to study some of these genes in a completely different environment. That was due to a typical EMBL feature, that is that we had joint lab meetings with Sarah Courtnage, and so I started working on tyrosine kinases. And so when Giulio left, and this is really one of those things that happen in life, and I think I was very lucky and blessed to have Thomas Graf believe in me. A year and a half into being a postdoc, he actually consulted with Lennart Philipson and Giulio Superti-Furga and Sarah Courtnage to give me an independent staff scientist. Imagine this, right? A year and a half into your postdoc, they admit you to faculty just because I have a big mouth and I don't know what other features I had. But Thomas is a clever person, and I think he was right, because we did assemble a great team, and it took us a long time to vindicate this trust. But what we did was elucidate some of the central features of cytoplasmic tyrosine kinase of the Sarc superfamily, and it turns out we were right almost in everything, despite what Maria said. So to the point that we made a patent with Emblem about a pocket that we discovered where myrstillation in ABL is regulating. And we had, we ended the Cell paper saying it has not escaped our notice that this is the pocket that would regulate it. And we kept the patent up for 2 or 3 years, but then at one stage, Gabor said, this is not worth it. Now, 22 years later, the best treatment in CML is through that pocket, a pocket discovered in this place and really discovered by these heroic people. And it's really Oliver Hanschel who did most of the work, who's now in Marburg. So the drug is called Semblix, and it's, I don't know whether there's EMBL in here, but it's called Semblix. And it's it will become first-line CML treatment based on a regulatory mechanism identified in Thomas Graf's department because of his generosity. Okay, but you learn everything in these years, right? You learn to be a human being, you learn to be a father. The first emails, the first everything, I really became a human being on this campus. This is the kind of support you get, these 4 Italian ladies supporting my early fatherhood with Giovanni here, who just turned 30. So I'm grateful to all the support I got. I think this lady gave a very good introductory talk, and I particularly liked the fact that the freedom of science is our competitive advantage. And this sentiment of freedom is really inherently part of the EMBL. And the rest of what I'm going to tell you has all to do with that. It was partly covered also by Maria. So for example, the first ever real poster that we gave as a group at EMBL is the 3-dimensional roboter, because here you had things that were rotating in the middle. And where else in the world would a group do as first ever poster something like this crazy? I can guarantee you that the industrialization of science that we're seeing now is not compatible with this degree of creativity. It just will not go down well. You'll have to be perfect. You'll have to be very well done. So this is what is possible at EMBL in those days. And I think this is, things like this, Photoshop, the photo lab that is glorious, taking pictures of this, again, things that you wouldn't do today. I'm not glorifying the past. I'm just telling you that the element of freedom that you had here with no police around. Actually, police not allowed. At least this was the credo, right? The police is not allowed. This is like paradise. So this was exactly the kind of sentiment that allows you to be really fearless in what you're doing. And this is what I'm trying to say. This is a headquarters for our community. There is no other place that is quite like this. It's extraterritorial. The police won't come. You may be strange anywhere else, but here you're at home. At least that's the way I feel. You can call it the Free Republic of Molecular Biology, and I think this is, in a way, that's what it is. And some sort of Hogwarts, or I apologize, this is really not my best art, but it is really inspirational. So what happens if you're carefree and fearless at EMBL? I'm not bragging with things that you shouldn't be doing at home, but just to give you a sense of it. When you're fighting with water every Friday afternoon with cell biology, one floor down, because they're having the cocktail, we're having a cocktail upstairs, we throw ice, we throw water, then Michael Way buys a big water bazooka, right? So what does Giulio do? In perfect, together with Thomas Graf, we unroll a hydrant fire hose. This is a professional thing that the fire police puts in your. We unroll it and carrying out 30 meters out of this place onto. You should have seen the face of the cell biology seeing this. It was so scary that Thomas Kraft here runs away. This is me. You can tell it by my ears. This is me, and this is Thomas running away because he was afraid that the hydrogen bomb will come because this is the halo of what we had been doing. Anyway, this is the sentiment, and this allows you to do a lot of things. And what I'm trying to tell you here is 3 micro stories that are connected with this sentiment of freedom and empowerment that you experience in this community. So the first one is this making of CeLSOM. You would not have done it in a different setting because it actually allowed us, at least in part, to be still involved in science. The quality of the science was guaranteed because there were 7 scientific founders. So it's really a beautiful story. And you see here a lot of the people that are still very important on campus. Gitte Neubauer, who's running GSK CeLSOM, myself, very important, and Claude Gavin, Per Borg, the who's who of the faculty then participated. A beautiful, beautiful adventure. And really, it's a success story because the community that did it, it's still there. And the amount of people that went through that company is truly amazing. So we are really thankful to EMBL for allowed to do this and so on. Bertrand Séraphin, who is not an unsung hero, he's a sung hero of this story, together with Matthias Wilm, had developed a method by which you could, in 2 steps, purify to almost stoichiometric quality kind of purification, complex that you analyze with mass spectrometry. And this was the basis for the company, and it allowed us to explore more and more. And it is really sort of that allowed us then to map the entire Cerevisiae interactome using this method. And so this was a beautiful work because we had to organize everything, and it was quite a few people are in the audience that will participate. An incredible experience to collaborate at that scale with many people. We had fantastic people on board, and then discover new principle, new concept, because people thought that there would be protein complexes, and some of them are, these days, people will say obvious, right? I mean, the ribosome and the nucleosome. But in those days, it wasn't that clear that there would be the sociology. Today, somebody is working, I saw sociology, molecular sociology. We invented that name. So this is all here. You can all read it up here. So we had the first chance. We were on the first page of the Frankfurt Allgemeine Zeitung, and Nature gave a press release just in a press conference just on this. And what did I do about this? Well, for sure, it was not easy to convince the management of a startup company that are putting millions in it to do a very, very basic science kind of project. And so one thing that I did is to convince this, and then together with Gitte to actually do it. But also I think it was very important. This is not protein, this is not individual protein, these are protein complexes. What's very nice was to sit down with Jörg Schulz, and a few other things, Manfred Bösch and Gitte, and start thinking about what it actually means to be able to do it at this scale. And so we thought, okay, maybe what it is really is protein complexes talking to each other. So it's sort of a cellular machinery. It was beautiful, and we really loved it. And then linking this to drugs. And to this day, this old cell-dome diagram still stands, where you say drugs actually operate on single targets, but those are linked into molecular machines that are by themselves also connected. So a great experience. Trick number 2. So it has to do with Kai Simon's T-shirt. So as it was mentioned already today, I was chair of the Alumni Association when we started thinking about what we should be doing at the next meeting and so on and so forth. And with Neovi Santama and Marie Ivanko Ruiz, they said, what if we had one of Kai Simon's T-shirt? You know, that he was wearing always special T-shirt, and they had one particular. If we had it, we would actually auction it and get some money for the association. This is how poor we feel every time. So you can leave your shirts behind here, we'll sell them. But the point is that then I had read a beautiful piece by Sidney Brenner that actually has said, we are not enough keeping records of the way we think, because the manuscripts are electronic now, and we don't keep the original versions, and so we don't really know how science evolves. And so what happened is that the light bulb went on in my head that says, okay, maybe what we should really be doing if we had Kai Simon's T-shirt is putting it in a museum, also known as archive. And this is how it all started. And you understand the elegance. This is why I'm wearing this jacket today, because I'm since then constantly aspiring to be as chic as Kai. This is on the day of the inauguration of the building next door in 78. This is the fantastic alumni board meeting that I had the pleasure to share. And this is Sidney Brenner, who instead of giving us one of his T-shirts, really supported us and said, I really think it is a great idea. And so what happened then is that through the work and Matthias was mentioned today a lot, Nush really incredible. She came to Vienna, we discussed it, and we implemented it. And then it took some time, but the archive was inaugurated some 8 years after. And now what it actually tells you is that many years after the foundation, 78, and here we're talking about 18, so it takes 40 years to EMBL to start actually an archive. So if you're making a new institute today, think about an archive immediately, because it will save you a lot of pain. Okay, trick number 3. This is the Casa Madre, this is the headquarters, this is Hogwarts. But then after Hogwarts, there is actually a place that is very nice. Probably state of the art for all of us is the Crick. So the Crick is in a nice London place, relatively expensive in the meantime, and it's done by Hawk Architects, and it was inaugurated 2016. So what can be better than this? You already know it, is a Crick in the sun. It's done by the same architects. It's going to be inaugurated 10 years after, so upgraded. It is 3 kilometers from the seaside, from the Mediterranean seaside in Sicily, so it's really very, very nice. And it consists of 2 chromosomes. And if you bear with me a second, I'll show you this is the city of Carini. This is the valley. This is the institute coming up. It will house some 600 people. These are the 2 chromosomes. They actually are facing the sea, as you will see in a minute. These are the hills. And so this is the train, and we will get a train station here. And so this is administration. This is a building devoted to teaching and also having other activities such as ethics and so on and so forth. And so this is really what we hope is going to be the Crick in the sun, and you're all invited. So this is another EMBL baby. Never, ever would I have dared to do something like this if I hadn't been trained in this place. Being fearless is something that you can only do if the environment that surrounds you teaches you that it's possible to do great things, because you're supported by a lot of people and a lot of colleagues. Palermo is here. This is the airport, and the new center is halfway through there. And we are absolutely happy if you would join us. It is a kind of baby of EMBL, also because it's inspired by the EMBL in terms of One Health. We heard fantastic talks today about this. It's going to be biomedical research considering the human as part of the environment. So the relationship between humans and environments will be very important. It's going to be as international as possible, depending on you. We will work on nutrition, pollution, infection, and many other things. And there is a foundation that is supporting this. I tell you this because as another EMBL project, I really hope that you will apply, we're looking for quite a few people. So to summarize, EMBL is a powerful idea. It's a reality. It's a community. What I've done and what I'm doing is a direct consequence of the confidence that is gained from experiencing this community operate. And I would like you to help me with this new one. And certainly the award that I'm getting here, I understand there's a license to do this. So thank you very much. And I finish here are the two gentlemen that have been very important in the years that I came here. And I finish saying, long live EMBL, long live the EMBL Alumni Association. Thank you very much.